All right, now we're gonna look at two different types of intrauterine transfers. So during in vitro fertilization, if you remember, what they do is they extract the gametes, the female and the male. So they grab the female eggs and the male sperm. They put it in a petri dish and then they transplant it in different locations, right? So that the locations of it is variable. But what's really important is when we look at the different styles of any kind of assisted technology for reproduction, any kind of assisted reproductive technologies. Now, we have two types, ZIFT and GIFT. So ZIFT represents zygote intrafallopian transfer, whereas GIFT is gamete intrafallopian transfer. So between these two, the difference is, so this little structure here, wonder what this is. This is the uterus, right? So you have the ovaries here, you have the fallopian tube presented here, and then you have the uterus. So I'll put uterus here in the middle. So we have the uterus. So you have the oviduct or the fallopian tube on both sides. And we're gonna compare the difference between ZIFT and GIFT. All right? So sperm and egg come out. We'll do the gamete intrafallopian transfer. They grab it, they put it on a petri dish. All right, so we draw a little petri dish here. So for the sake of color, we'll put white as sperm. And then we have some eggs in pink. So we'll have some eggs here, some eggs here, and they're all cultured on a petri dish. However, what's really important about gamete intrafallopian transfer is that there is no fertilization that takes place. They're only removed, put on a petri dish for some kind of selection, and then reintroduced back into the fallopian tube. So again, if I were to draw it, so you have a couple eggs here, and then we have a couple sperm here. And this is an example of gamete intrafallopian transfer, where there's no fertilized egg, so there's no zygote. And so the separate gametes are transferred in the fallopian tube, waiting for fertilization. ZIFT, however, zygote intrafallopian transfer. So again, I draw the petri dish here. Egg and sperm are removed. But what's important now is when the sperm and egg are transferred onto the plate, they're left there longer in order to be fertilized. So now, if I do it in green, we'll call those zygotes. Zygote is what? A fertilized egg. So when the sperm and egg combine, become diploid, they become this structure called the zygote. And so now these zygotes are transferred into the fallopian tube. So that's the difference between ZIFT and GIFT. So GIFT happens separately, so the gametes are separate before they enter the fallopian tube, whereas ZIFT, the sperm and egg combine in vitro, so outside. So let's write that. So in vitro fertilization. This is an example of in vitro fertilization. And so then that fertilized egg or that zygote gets transferred to the fallopian tube. So these are the two different styles of intrafallopian transfer.